All right, hello everybody. This week is week 7, and this week I will be talking about soil microbiology and biochemistry. So to get started off, I want to link back to our previous unit, which is about soil physical properties. We talked a lot about bulk density and porosity, and one of the things that is a result of bulk density and porosity is aggregation. So aggregation is a component of soil structure, and it's made up of aggregates. These are kind of the building blocks of soil structure. And aggregates are just clumps of soil particles. So what happens is you get kind of soil carbon inputs here, and it creates these aggregates by acting like glue. It kind of glues together all of these different particles. So we have different size aggregates. So here we have a nice little illustration where you can see this is what the silt and clay particles are. And then we have some microbes in there too. And so a microaggregate here, it's about from 0.053 millimeters to 0.25 millimeters in size. And these microaggregates are built up of these silt and clay particles and microbes, and it's all kind of glued together with soil carbon. Then those come together and they form what's called a macroaggregate. Now, macroaggregates are 0.25 to 2 millimeter size clumps. So macroaggregates, you can see there's a bunch of microaggregates within it. And then these green and red lines here are fungi and organic matter that's kind of in there. And here, this is a big root kind of coming right through. So this is where the soil carbon inputs come from. And as this picture kind of implies, you see microbes all over in here in the aggregates and whatnot. These aggregates are basically little microbe homes, microbe hotels, if you will. So they're living in here, doing their microbe thing. So that's, they're kind of like little microbial hotels. So, but who's actually in there? Well, let's go back to the kind of intro biology approach here. There are 3 domains of life, and these are all within soil. So we have here bacteria, and again, bacteria, if you remember, are prokaryotes, are single-celled organisms with phospholipid bilayers. So these are fats with a phosphate head kind of attached on there, and there's 2 layers of them. So that's a characteristic of bacteria. Now, Archaea, on the other hand, here are prokaryotes, so single-celled organisms, and they have a similar phospholipid layer, but they only have 1 layer of these phospholipids around the edge of their cell. So slightly less complex organisms there. Then we have eukarya, which are multi-celled organisms, and specifically they have membrane-bound organelles within them. So eukarya here, which is just one of these 3 domains, is all of these multi-celled organisms that we see every day. It's you, it's me, it's your pet chameleon, it's plants, it's all of those things are in this domain of eukarya. Now, traditionally, we've thought of life as being in 3 domains. Recently, there has been some kind of discussion on this, and it all kind of revolves around what's called last universal common ancestor. So last universal common ancestor is where this phylogenetic tree breaks off. So you see here that bacteria and archaea and eukarya kind of broke off a long time ago here, phylogenetically speaking. And then archaea and eukarya split at some point due to this last universal common ancestor. However, as I mentioned, we're starting to kind of reconsider this. There's an important soil microorganism that is really influential in this. And it's telling us that, hey, maybe we actually have a 2-domain system, that this 3-domain system might be outdated. And instead we have 2 of these domains. So bacteria kind of all by itself here. And then we have archaea. So most importantly, you see that eukarya is no longer distinct from archaea, but instead it's kind of a branch off of this. And again, they use this last universal common ancestor approach here to say, oh, there's actually a microbe here, an Archaea here, that kind of links these two together, that is, yeah, kind of bridging that gap there. And that's a soil microbe. If you want to know more about that, take soil microbiology with me next semester and we'll talk all about it. So maybe just to get a handle on what these things look like here, here's some pictures of these soil microbes that have been colorized. These are scanning electron microscope pictures. So here's a bacteria here, this blue and yellow picture, and it's on a root right here. This pink stuff is a root, and it's attached on there. Over here we have eukarya, specifically fungi. And that fungi, you can see it's kind of got this net everywhere. Over here we have another eukarya, which is also a fungi, but this is a very specific type of fungi. It's Penicillium. Now, Penicillium, you might hear sounds like penicillin, which is because penicillin is an antibiotic, and it's one that was derived from a soil microbe here, from these spores. Now, in soil, these kind of different organisms, these different types of organisms are not so isolated from one another. They're all kind of happening all at once. You can see here we've got bacteria and fungi all kind of mixed together on the soil surface here. So they're all in there playing together. Now, when we talk about bacteria, there are really two main types of bacteria, and we define these two types of bacteria based on a Gram stain technique. So what the Gram stain technique does is it adds a dye, and that dye will get absorbed within the bacterial cell walls. So when only a little bit of that dye gets absorbed, we call it Gram negative. And when a whole bunch gets absorbed, we call that Gram positive. Now, what that dye is actually getting absorbed to is something called Peptidoglycan. So these are big molecules, these are polymers, and the dye gets stuck to those. And so we can look at it and see, that that we have Gram positive or Gram negative. Now, this thick peptidoglycan wall here for Gram positive bacteria means that it's a little more protected. It's a lot of material out here, and it actually makes them more resilient to things like drying, for example. Similarly, when we talk about soil fungi, there are three types of fungi, but only two of them are really common in soil. So the first one is called Ectomycorrhizal fungi. Now, ecto means outside, and so ectomycorrhizal associates with plant roots here. And what it does is ectomycorrhizal will be attached on the outside of the plant root. Now, it also invades the plant root a little bit, but it's still, as you can see here, outside of these cells. It's on the interstitial space between cell walls. So it's still outside of all these things. But it develops this thing called a Hardig net, which is a kind of, it's a net of this fungi, and it then puts these hyphae out into the soil, bulk soil, and it extends the total surface area that a root can then reach. So it's beneficial because fungi don't need carbon, so they get it from their sugar daddy, the plant, and that is this root here. And then they, in return, bring nutrients to the plant root by extending into this bulk soil. Now, the other type of fungi is called endomycorrhizal fungi, and endo meaning inside. Endomycorrhizal fungi are also known as arbuscular fungi. Now, endomycorrhizal fungi, just like ectomycorrhizal, need that carbon, need that sugar, but they go about it a little differently by actually entering the plant root. So what they do is they invade here, and they develop these things called arbuscules. And as you can see here, these arbuscules are within the plant cell. And so because it's within the plant cell, there's actually a more rapid exchange of nutrients between these two. It's like being on the freeway here as opposed to being on side streets when you're driving. It's just much quicker. And so then when these hyphae are out here and extending into the soil, the nutrients can be exchanged more quickly. Now, these are just some pictures real quick that you can see from these electron microscopes. So here you can see exactly what that Hardig net looks like. It's just coating the root here. You can see it's kind of all over it. It's really dense. And then over here, you can see kind of the arbuscules. You can see that it's inside this plant cell here. You can see it kind of looks like a little brain or something like that, but it's all infected in this plant root cell. And so it really does exchange nutrients really rapidly for them. You can see the highway, if you will, here. Another important group of soil microbes are the actinomycetes. So actinomycetes are kind of this bridge between bacteria and fungi. They're technically a bacteria, specifically a gram-positive bacteria, but they kind of have this fungi type characteristic, which is they form that hyphal net. You can see here that they have this really web-like look. And so because of that, they're, functionally kind of similar to both bacteria and fungi and, deserve their own special call out here. Now that we know what microbial houses are and we know kind of who they are, the next thing is we want to know what they eat. So just a little quick discussion on microbial metabolism. So when we talk about metabolism, it's these opposing processes of anabolism and catabolism. So anabolism is over here, and anabolism is essentially biosynthesis. We take monomers, which are smaller molecules, and we build larger macromolecules out of them. Now, that costs energy, which you see here with the ATP, the adenosine triphosphate, and it costs them energy to build those macromolecules. So that's anabolism. On the other side of this coin is catabolism, where you take resources from the environment and you break them down, which gives you energy here, right, your ATP, but you break them down into molecules. So this is how you get energy to do the things like running, jumping, or whatever it is that microbes get up to. So it can be kind of difficult to remember which one is which when we talk about anabolism and catabolism. So the way that I like to think of it is I like to think of my cat. Now, my cat, like many other cats, has a destructive nature about her. And whenever something is on my counter, she likes to push it off and break it because she is a cat. Similarly, think about catabolism as breaking things down, just like my cat when she sees my wine glasses. Now I want to do a little spotlight within catabolism, or should I say catabolism, a little spotlight on oxidation reduction reactions, which is back to your kind of general chemistry class. Now, I know when I took general chemistry, I was not so fond of redox reactions, just like Grumpy Cat here. And so I want to just do a quick review. They're a little confusing, and hopefully I can just give you the basics again, review the basics and how they relate to soil microbial metabolism. So here on the left-hand side, we have the reactants. So we have reactant A and reactant B, and these have no net charge on them. They have 0 net charge. Now, they undergo this redox reaction, which is 2 half reactions. And we remember these 2 half reactions using the acronym OIL-RIG. So oxidation is losing an electron, and reduction is gaining an electron. There's also Leo goes Gur, so it's losing an electron is oxidation, and gaining an electron is reduction. Now, what happens is those reactants here become these products. Now, for example, if you look at reactant A, it now is over here, and it lost an electron, so now it's a net positive charge. It's also lower energy now. It donated its electron, so it has less energy. However, the product B here, right, it gained an electron, so now it has a negative charge, but it also has more energy, right? Electrons are energy, as anyone who uses electricity might know. So similarly, this is how we get energy from chemical bonds. Now, that process is a little more complicated. It doesn't just happen spontaneously, because we have our reactants where that energy is stored in those chemical bonds, and we have our products here. And the difference between the energy level of those reactants and products is the amount of energy that we can get. Now, between those two is this hill here, this energetic hill. You have to pay a little bit of energy to break those bonds, and then you get more energy than you paid, but you kind of got to get over that hump there. Now, that little energetic hill, we need a catalyst to kind of get this reaction started, if you will. And so those catalysts are called enzymes. So enzymes aid in the breakdown of these reactants into these products and help us get energy. So when we think about those redox reactions, we want to be able to look at the reaction and know which of our reactants is going to give up an electron and which one is going to accept the electron. And the difference between those two is called the reduction potential. Reduction potential is directly related to the amount of energy that results from that reaction. Now, a better electron donor has a negative value of this E prime or reduction potential, and a better electron acceptor has a positive value. The greater the difference between those, the greater the amount of energy that you can get from that reaction. So because there's, the amount of energy you get from it is dependent on how good of an electron donor and acceptor you have, there's kind of this preferential order, right? Some compounds are going to be better donors and acceptors, and therefore, from the microbial perspective, you don't want to do any more work than you have to get the most energy. So we have something called the redox ladder. And that redox ladder, when we apply it in the real world, there's a perfect analog within soil, so specifically within flooded soils. So I'll explain why in just a second here. So when we have that reaction, the two things we need to know are the electron acceptor and the product. So the most ideal electron acceptor is oxygen, okay? So oxygen being O2, it accepts an electron and it ends up producing a molecule of water. So that's an aerobic reaction in the process or in the presence of oxygen, and that's undertaken by aerobic bacteria. Now, sometimes we don't have enough oxygen to accept those electrons, and so the next thing down the line is nitrate. And that nitrate gets, when it accepts electrons, it ends up as N2 gas. And that process is known as denitrification, and it's undertaken by denitrifying bacteria. When we're out of nitrate, we are looking then, our next terminal electron acceptor is manganese, and it's reduced to manganese 2+. And again, that's manganese reducing bacteria. Similarly, then we have iron, which can go from an Fe3+ here to an Fe2+, and that's undertaken by iron reducing bacteria. Up next is sulfate. So this one is producing H2S gas. This is sulfate reducing bacteria. So H2S gas is, if you ever smell that rotten egg smell, that's what's happening here, is this sulfate is getting converted to that. So this is the rotten egg gas. And then lastly, you have CO2 is eventually your final electron acceptor, which gives us methane, and that's undertaken by methanogens. And methanogens are almost always Archaea. And so what happens is here, we go down this electron or this redox ladder. So if we don't have oxygen, we get nitrate, manganese, iron, sulfate, and then CO2. And so this ladder, to remember the order of that, we use the mnemonic, "Oh no, my feet so cold." And the way I like to think of that is it happens in anaerobic soils, like, so say if it's flooded, right, all the pores are full of water, so there's no air. So then we start going down this redox ladder. And when you step in a flooded soil, your feet get wet and cold. And so what would you say? Well, oh no, my feet so cold. Now, microbes don't want to go through that redox ladder. And why don't they want to go through that whole thing? Because as you go down that redox ladder, you get a decreasing amount of energy from each mole of reaction that happens. So the most common of these redox reactions that happens is called aerobic respiration. So we do this when we are out. If I go for a run, I am burning C carbon. So this is like a carbohydrate or a sugar of some sort. And I'm breathing in oxygen as I gasp for air. And then those two things meet, and that sugar donates an electron, and that oxygen accepts it, and I breathe out CO2, and I produce a little bit of water. So similarly, when microbes are working hard, they are going to be consuming sugars, accepting electron, or using oxygen, which is accepting the electron, and breathing out CO2. So this reaction is built up of two half reactions. So the first of the half reactions is we have carbon in its zero oxidation state, which leads to one carbon in a +4, and that means it's giving up 4 electrons. Now those 4 electrons get combined with 2 oxygens in its zero redox state, and those then become, both of them become -2, so we have 2 O2- molecules. So these two things happen, and then you see that that oxygen here, that's a -2, combines with those 2 H2s to produce water. And that carbon is now -4 and combines with oxygen as well. And now remember, when we go through that, right, there you have the reactants, and it gets oxidized, and the products, the difference between those energy levels is the amount of energy that gets released. So for example, if I was going for my run, this is the amount of energy that gets released so I can run. So to do a quick example of tying it together, microbes are, they like the carbon. And so if we want to store carbon, we want to keep the carbon away from microbes. So microbes are often carbon limited. That's usually the most limiting nutrient for them. So those carbohydrates get combined with oxygen and they breathe off CO2. Now there's a couple of ways that microbes can get at soil carbon, but one of them is to circle back to those aggregates that we talked about, their home. So when aggregates break open, it exposes soil carbon, right? So those aggregates were clumps, and when those clumps break apart, all of a sudden microbes can access the soil carbon. Now, when they start eating up that soil carbon, some of it gets incorporated as biomass. It becomes part of their body, right? But most of it, over 90% of it, gets respired as CO2. Now, this might sound like a very small micro process, but when we start looking at this at the global scale, you can see that it has some really big repercussions. So up top here is the amount of soil organic carbon, the amount of carbon in the soil in the top 5 centimeters on a global level. Now I want to direct your attention to a couple specific places. Up here in the higher northern latitudes, we have lots of soil carbon. But then some places like the Sahara Desert, for example, we don't have as much. But up here at the top, we have lots. Now, let's compare those places with the places that we have respiration, or as they call it, heterotrophic respiration. And so that respiration, if you look at the places that have low respiration, a lot of those are also at the higher latitudes. And so on net, because they have lots of carbon in the soil and not much being respired, we can, that's a cause and effect here. The microbes aren't respiring this stuff up here. You can see that this is also the colder climates. Similarly, when they don't have much to eat, like in the Sahara Desert, for example, then there's also not much soil carbon there. When we kind of aggregate that, or put it together rather, and we look at it across latitudes, we can see that this makes for some huge amounts of carbon stored in the soil. So here is carbon stocks. So this green stuff, that is the above ground carbon, the amount of carbon that is above ground in things like plants, humans, animals, etc. Now let's just compare that to the amount of carbon that is below ground, so soil organic carbon at varying depths here. Now you see that first, there is a lot of carbon in these higher latitudes, so towards the North Pole. So you can see that here, this is about the 30 centimeters right here, but you can see that below 30 centimeters are just astronomical amounts of carbon. And that carbon, is right now not exposed to microbial attack because it's frozen, the soils are waterlogged, so there's no oxygen, so microbes can't get to it. So you can see here that microbes, similarly, in the top 30 centimeters, there's not, that's where most of microbial biomass is. From the 30 to 100 centimeters, it's pretty minuscule amounts here. And that's because they don't have access to this enormous amount of carbon below 100 centimeters here, all of this. So the next thing that I want to show you is just how much of that is at deeper depths, both at the equator here in the tropical regions, temperate regions, which is about where we are here, and then in these northern latitudes, there is so much carbon that is stored here. So when we talk about things like climate change and as soil warms, so as that soil warms and that carbon becomes accessible to soil microbes, they will eat it up and they will respire it, right? 90% of it gets respired and sent into the atmosphere. So when we talk about these redox reactions and microbes being hungry and carbon limited, this has some pretty profound implications for global carbon storage and climate change, et cetera. So just a quick review. There are three main types of soil microbes. So we have fungi, bacteria, and archaea. Now, fungi, we have two main types in soil. The first is endomycorrhizal, endo meaning within the soil or within the plant root. These are also known as arbuscular mycorrhizal fungi. So again, they invade the plant root cells with little arbuscules, and they're literally in that plant cell. Ectomycorrhizal are outside, so ecto meaning out, and so those occupy the spaces between those plant root cells, and they make this huge net on the surface of plant roots. Bacteria, there are a couple kinds we want to think about. So there's gram-positive, which they have a thick external layer of peptidoglycan that holds a gram stain. Gram-negative have a thin internal layer of the same thing, peptidoglycan. And then we did a little spotlight on actinomycetes, which are a gram-positive bacteria that produce hyphal nets, just like fungi. Now, fungi and bacteria, they all have metabolism, and metabolism is just the combination on net of anabolism plus catabolism. Catabolism, again, like cats, likes to break things down. And one example that we used was redox reactions. Now, redox reactions are oxidation and reduction. And remember, oil rig. And that reaction needs to get over an energetic hill, if you will, in order to get the energy that is from those chemical bonds. Now, to get over that energetic hill, we have enzymes, which are catalysts for that. Now, deciding how much energy we get from those redox reactions, we have terminal electron acceptors. Oops, typo here. It says election, but it means electron acceptors. And so those terminal electron acceptors, we have a redox ladder, and we have an order of preference of those. So the mnemonic that we use to remember that is, oh no, my feet so cold. We have oxygen as the most preferred, then nitrogen, manganese, iron, sulfate, and then CO2 as our least preferred electron acceptor. And so with that, I will say thank you, and I will see you all in class this week. Bye.